95% of the living space on this planet lies in permanent darkness, and in that darkness, evolution has been running an experiment with no witnesses, no mercy, and no obligation to produce anything we would recognize as an animal. Down there, pressure doubles and redoubles with every 100 meters until it could crush steel. Sunlight dies before it reaches 1,000, and food arrives so rarely that a single wasted movement can be fatal. So life has answered with weapons instead of comfort. Bodies made of glass that erase their own shadows, reef dwellers armored like war machines and laced with poison beneath their ornamental skin, hunters that grow their own light and dangle it as bait, gelatinous specialists that survive 1,000 atmospheres by surrendering everything solid inside them, venom refined into a precision instrument, and ancient predators whose blueprints have not needed revision since before the dinosaurs vanished. What follows is a descent through 3 biological zones, from the sunlit reef to the crushing black of the abyssal plain, in search of the strangest survivors ever catalogued, and the uncomfortable question of how many more are still down there, unnamed, in water no human eye has ever entered. "95% of the living space on this planet lies beneath the reach of sunlight, in water so dark that eyes become optional, so cold that metabolism slows to a crawl, and so heavily pressurized that a human body would be crushed into unrecognizable tissue within seconds. This is the largest habitat on Earth, and it is also the least observed. Every square kilometer of it functions as an unsupervised experiment in survival, running continuously for hundreds of millions of years, with no observer, no witness, and no mercy for failure. What emerges from that experiment is not beautiful in the way a lion or an eagle is beautiful. It is functional, ruthless, and frequently so strange that the human eye struggles to interpret what it is seeing. The ocean is nature's most unforgiving laboratory precisely because it offers so many ways to die and so few ways to hide. There is no cave to retreat into on the open abyssal plain. There is no tree to climb, no burrow to dig, no season to wait out. In the water column, an animal is suspended in 3 dimensions, exposed on every side, visible from below as a silhouette and from above as a shadow. Under those conditions, evolution does not produce compromise. It produces specialists so extreme that their anatomy has been rewritten from the inside out, their bones dissolved into cartilage or gelatin, their organs reorganized around a single overwhelming priority, whether that priority is concealment, ambush, or the simple refusal to starve. What follows is a descent, not a wandering survey, but a structured fall through 3 distinct biological zones, each one demanding a more alien solution to the problem of staying alive. It begins in the sunlit shallows, in the coral reefs and coastal shelves where light still penetrates, and where competition is at its most savage because everything can see everything else. Here the strangeness is disguised as decoration. Venom hides inside patterns that look ornamental. Ambush predators wear the texture of stone. The reef is the most crowded neighborhood in the sea, and in crowded neighborhoods, weaponry becomes a requirement rather than an advantage. Below that lies the twilight zone, the mesopelagic band beginning around 200 meters down, where sunlight fails by degrees until only the faintest blue remains, and then nothing at all. This is the zone of the Great Vertical Migration, the largest coordinated movement of animals on the planet, in which countless tons of biomass rise toward the surface each night to feed and sink back into darkness before dawn. It is a zone of half light and half certainty, where camouflage strategies fail because there is no background to blend into, and where evolution has responded by making bodies disappear entirely. Transparent flesh. mirrored flanks, skin that absorbs light rather than reflecting it. Here, being invisible is not a trick, it is anatomy, and then, deeper still, the abyss. Below 1,000 meters, the water is permanently black, permanently near freezing, and permanently under a weight that surface intuition cannot process. Food arrives as scraps drifting down from far above, the slow rain of dead plankton and fish scales and larger carcasses that fall through the water like snow. Encounters are rare, meals are rarer, and in a world where a predator may only meet prey a handful of times in a year, the mechanisms of capture become extreme because a single missed strike can mean starvation. This is where the ocean stops producing animals that look like animals and begins producing something closer to machinery, the force that shapes all of it. The invisible sculptor behind every one of these forms is pressure. At the surface, the atmosphere presses down at roughly 1 bar. Descend just 10 meters into seawater, and that pressure doubles. Every additional 10 meters adds another atmosphere, relentlessly without pause, so that by 100 meters an animal carries 11 atmospheres, by 1,000 meters more than 100, and at the bottom of the deepest trenches nearly 11 kilometers down, the load exceeds 1,000 atmospheres. That is more than a ton of force pressing on every square centimeter of flesh. Gas-filled cavities collapse, rigid structures fracture. Proteins, the delicate folded machinery of life itself, begin to deform and lose their shape under that kind of squeeze. So the deep sea rebuilt its inhabitants from the molecule upward. Deep-dwelling fish accumulate specialized compounds in their tissues that stabilize proteins against the crush, chemicals present in such concentrations that they set a hard physiological ceiling on how deep any fish can survive, somewhere around 8,200 meters. Below that line, no fish exists anywhere on Earth, not because the water is too cold or too dark, but because the biochemistry simply fails. Swim bladders, so useful in shallow water, vanish or fill with lipid instead of gas. Skeletons thin, muscles become watery and pale, bodies become soft, flexible, and largely incompressible because water itself does not compress, and a creature made mostly of water has nothing left for the pressure to crush. The result is anatomy that seems flimsy, even pitiful, when hauled to the surface, yet is perfectly engineered for a world that would reduce a submarine hull to scrap. From that single physical gradient, 3 great categories of adaptation emerge, and they recur again and again at every depth like variations on a theme. The first is transparency, the most radical form of camouflage in the natural world. Where there is nothing to hide behind, the only solution is to stop existing visually altogether. Certain squid, larval fish, and open water crustaceans have reduced their bodies to clear tissue and refractive fluid, their internal organs shrunk, silvered or oriented edge-on to hide from eyes below. Some deep-sea creatures have taken the opposite path and evolved skin so black that it absorbs more than 99.5% of all light that touches it, rivaling the darkest materials ever manufactured by human engineers. Whether by vanishing into clarity or vanishing into shadow, the goal is identical, and it is not modesty. It is the difference between eating and being eaten. The second category is armor, and in the ocean, armor is rarely purely defensive. Shells become clubs, spines become injection systems, plates become the anchor points for muscles that can accelerate a limb faster than a bullet leaves a rifle. Some crustaceans have mineralized their exoskeletons with iron compounds. Some mollusks have turned their shells into launch platforms for harpoons loaded with neurotoxin. In an environment where soft bodies are the norm, hard structures become opportunities for offense, and the line between protection and predation blurs until it disappears entirely. The third category is light. In perpetual darkness, an animal that controls light controls everything. Bioluminescence occurs in an estimated 3/4 of the creatures inhabiting the open ocean's mid-depths, produced either by the animal's own chemistry or by colonies of bacteria kept alive in dedicated organs. It is used to blind, to distract, to counter-illuminate a belly so a silhouette disappears against faint light from above, and to signal across black water to potential mates that may never be encountered again. But most chillingly, it is used as bait. A glowing lure suspended in absolute darkness is an offer that a starving animal cannot refuse, and behind that light waits a mouth built for a single, irreversible closure. None of these mechanisms were designed. Each was forged, tested against extinction, and retained only because the alternatives died. What lies ahead in this descent will test more than intuition about anatomy. It will test the categories themselves. There are creatures in the deep whose evolutionary position remains genuinely contested, animals that were classified, reclassified, and then argued over again as new specimens surfaced with features that belong to no known lineage. There are species known from a single damaged carcass, hauled up decades ago, never seen alive. There are entire body plans so reduced, so specialized, so far removed from any relative that molecular analysis has produced results biologists initially refused to believe. Some are so bizarre that their males and females were cataloged as separate species belonging to separate families, a mistake that stood uncorrected for years. The ocean does not owe us comprehensible organisms. It has been generating solutions in the dark for far longer than vertebrates have existed on land, and it has not been checking its work against human expectations. So the descent begins in bright water among creatures whose strangeness is still recognizable, and it moves downward, atmosphere by atmosphere, toward forms that push at the edges of what the word animal is meant to contain. "600 meters beneath the surface, a hunter hangs in the water column with its arms folded above its head, and there is almost nothing there to see. A remotely operated vehicle's lights sweep across it and pass through it. What returns to the camera is a faint outline, a pair of dark ovals that might be eyes, a small dense mass that could be a swallowed meal, and a shimmer of edges where one clear substance meets another. This is a glass squid, a member of the family Cranchiidae, and it represents one of the most complete acts of disappearance that biology has ever produced. There are roughly 60 described species in the family, ranging from animals a few centimeters long to giants that would take up an entire room, and nearly all of them share the same defining trick. They have solved the problem of being visible by refusing to interact with light at all. Transparency is not the same as camouflage. Camouflage is imitation, a matching of colors and patterns against a background that may change the moment the animal moves. Transparency is subtraction. Instead of resembling the surrounding water, the glass squid becomes optically indistinguishable from it, and it accomplishes this through a combination of chemistry and structural engineering that human material science has only recently begun to reverse engineer. The tissues are built to have a refractive index almost identical to seawater, which means light passing from water into flesh and back out again barely bends and barely scatters. Most animal tissue is a chaos of membranes, pigments, mitochondria, and collagen fibers, each with slightly different optical properties, each one a tiny surface for light to bounce off. The glass squid has stripped that chaos down. Its musculature is thinned and reorganized, its connective tissue rendered clear, its body cavity filled not with dense organs, but with a vast chamber of buoyant fluid rich in ammonium chloride, a solution lighter than seawater that lets the animal hover without swimming, and without the gas-filled bladder that pressure at these depths would crush. The problem is that transparency is never total, and the survivors are the ones that solved the remainder. Two features cannot be made clear. The first is the digestive gland, a dense, opaque organ that no evolutionary trick has managed to render invisible. Glass squid deal with it by orienting it vertically, keeping the narrowest possible profile aimed toward the faint light filtering down from above so that the shadow it casts is reduced to a thin needle. The second problem is the eyes. Vision requires pigment because pigment is what absorbs light and converts it into a signal, and pigment by definition is visible, so the eyes remain as two dark points suspended in an otherwise absent body. And the glass squid compensates by mounting photophores directly beneath them, light-producing organs that shine downward with a soft glow calibrated to erase the eye's own shadow. It is counter-illumination applied not to a whole animal, but to a single unavoidable flaw in an otherwise perfect vanishing act. Related transparent hunters have gone further still. Certain hypereid amphipods, tiny clear predators of the twilight zone, carry across their exoskeletons a layer of nanoscale bumps and microscopic sphere-like structures that behave as an anti-reflective coating, cutting surface reflections by a factor that in some measurements approaches 100-fold. They are not merely clear, they are engineered not to glint. All of this exists for a reason, and the reason is a band of water between roughly 200 and 1,000 meters known as the mesopelagic or the twilight zone. It is the largest continuous habitat on the planet and the single most important battleground in the ocean. Here, sunlight has not yet failed completely, but it has been reduced to a dim blue residue falling from directly overhead, enough to see by and far more dangerously, enough to be seen by. In the blackness below 1,000 meters, an animal's silhouette means nothing because there is no background light to be silhouetted against. In the bright shallows, there is nowhere to hide at all. But in the twilight zone, the two cues that betray a body are shadow and silhouette, and an animal that eliminates both has effectively left the visual world. That is why transparency reaches its evolutionary peak precisely here. Among glass squid, glass octopuses, transparent snails, clear-bodied worms, larval eels that resemble sheets of cellophane with a spine, and juvenile fish whose organs are the only part of them a predator can find. Among the strangest of these is the glass octopus, Vitrilidonella richardi, a species known primarily from damaged specimens hauled up in nets and observed alive on only a handful of occasions. In 2021, researchers working in the remote waters near the Phoenix Islands filmed one twice in the space of days, and what the footage revealed was an animal reduced to a suggestion. Nearly everything visible belonged to the optic nerves, the eyes, and the gut. The rest was outline. The hunting behavior of these animals overturns almost every expectation about what a predator looks like. There is no chase. There is no burst of speed. Food in the mesopelagic is scarce and unpredictable, and every calorie burned in pursuit is a calorie that may not be replaced for days, so the glass squid has traded velocity for stillness. It positions itself in the water column, sometimes head down, sometimes with its arms drawn up into a cone above the head, and it waits. Its ammonium-filled body makes it very nearly neutrally buoyant, which means holding position costs it almost nothing. Hours can pass with barely a fin beat. Fine sensory hairs along the body register the pressure waves of anything moving nearby, and because the animal itself is effectively invisible, It does not need to conceal itself behind rocks or in sediment. It hides inside open water. Small fish, crustaceans, and other squid drift past the shape they cannot resolve until they are already within reach, and by then the decision has been made for them. The strike is where the illusion ends. Cephalopod arms come equipped with far more than suction, and in the cranchids, the feeding tentacles are lined with suckers that in many species are reinforced with rings of chitin, and in others replaced outright by curved hooks. When prey enters the cone of arms, the tentacles snap inward and the hooks anchor into flesh, converting a passive drifting object into a cage with barbs. What follows is not a struggle of strength but of anatomy. The prey is drawn toward the center of the arm crown, where a hard, parrot-like beak waits, capable of shearing through muscle and bone in a series of precise bites. Behind the beak sits the radula, a rasping band of teeth that shreds tissue into fragments small enough to pass down a narrow esophagus that in squid runs directly through the brain, a structural quirk that makes large, hasty swallowing physically impossible and turns every meal into methodical dismemberment. Many squid also deliver a paralytic component through their saliva, cephalotoxins that immobilize prey and begin the process of breaking it down before it has been fully consumed. And when the glass squid is the one being hunted, its defenses invert its own transparency. Chromatophores, pigment cells scattered through the otherwise clear skin, can be expanded in an instant to flush the body with color, an abrupt appearance designed to startle. Some species can release ink into a chamber within their own translucent body, so that the animal fills with a spreading darkness and then jets away from the cloud it has just become. Others fold their arms and pull their bodies into a compact ball, tucking every exposed edge inward until there is nothing left to grip. The final escalation in this family is a matter of scale, and it is worth stating plainly. The colossal squid, Mesonacuteuthis hamiltoni, the heaviest invertebrate known to science, an animal that may exceed 6 meters and half a ton, and whose eyes are the largest of any creature that has ever lived, is a cranchid. It is a glass squid. Its juveniles drift through the twilight zone as clear, near-invisible specks, and as they grow, they abandon transparency for a different kind of dominance, arming the clubs of their tentacles with dozens of swiveling three-pointed hooks that rotate to catch flesh from any angle. The same lineage that perfected disappearance also produced the largest ambush predator in the deep, and the logic never changed. Do not be seen. Do not move. Let the ocean deliver, and then close. Rise 1,000 meters and the rules change. Light returns, and with it color, structure, competition, and violence at a density found nowhere else in the sea. Coral reefs occupy less than 1% of the ocean floor, and yet shelter perhaps a quarter of all marine species, which means that every square meter of that living limestone is contested ground. Here, transparency is useless. In water this bright, against a background this cluttered, an animal cannot vanish into open blue. It must instead choose a different answer to the same question that governed the twilight zone, and on the reef that answer is frequently armor, not the soft, water-filled bodies of the deep, but hard cases, spines, plates, and fused bone wrapped around creatures that look less like fish and more like ornaments, and which conceal within their decorative shells a chemistry capable of killing everything in a confined body of water. The clearest expression of that strategy belongs to the boxfish, the family Ostracidae, roughly 24 species scattered across tropical reefs in the Indian, Pacific, and Atlantic Oceans. Their name is not a metaphor. A boxfish is, in structural terms, a fish inside a box. The scales that in most bony fishes remain separate overlapping and flexible, have instead fused into rigid hexagonal plates called scutes, locked edge to edge into a single continuous shell known as a carapace. The result is a triangular or square-sided casing enclosing the entire body, with only a handful of openings cut into it, two for the eyes, one for the small protrusible mouth, gaps for the gill slits, and holes through which the fins and tail emerge. Everything else is sealed. Unlike almost every other fish on the reef, a boxfish cannot bend its body. The spine is effectively immobilized within its own armor, the ribs reduced or absent, and locomotion delegated entirely to the appendages that stick out of the shell. That armor is not decorative. The hexagonal scutes interlock along sutured margins, and beneath each plate lie collagen fibers arranged in layered sheets that anchor into the connective tissue below. The geometry matters. When a predator's tooth or jaw applies force to one plate, that force is transmitted outward across the sutures and distributed through neighboring scutes rather than concentrating at a single point. Engineers who have studied boxfish carapaces describe a structure that resists penetration while retaining a controlled degree of give, absorbing energy instead of shattering. For a fish the length of a human hand, this is armor that meaningfully changes the calculation for a grouper or a snapper, converting an easy mouthful into a hard, awkward, unswallowable object that damages the mouth attempting to crush it. Some relatives push the geometry further still. The longhorn cowfish, Lectoria cornuta, grows a pair of forward projecting horns above its eyes and a second pair angled backward from the base of its shell, four bony spikes arranged so that any predator trying to take the animal head on or tail first encounters a shape that will not fit down a throat. The cost of all this is movement, and the cost is severe. A boxfish cannot undulate, it cannot sprint. It swims by ostraciform locomotion, sculling rapidly with its pectoral fins, while the dorsal and anal fins contribute short bursts of thrust, and the small tail is deployed mainly as an occasional paddle, an oar rather than an engine. The effect, watched from a distance, is almost comic. A rigid, brightly patterned cube hovering above the coral, fins whirring like tiny propellers, drifting with the deliberate slowness of something that has never in its evolutionary history needed to flee. For decades, the prevailing explanation was that the carapace itself provided passive stability, its keels generating self-correcting vortices that kept the fish upright without effort, an idea attractive enough that automotive designers built a concept car around it. More recent work using physical models in flow tanks overturned that reading. The carapace is not self-stabilizing at all. The vortices it sheds are destabilizing, amplifying deviations rather than damping them, and the boxfish holds its position through constant, precise, high-frequency fin corrections. What the shape actually delivers is not steadiness but maneuverability, the ability to pivot on the spot, reverse, hover motionless in surge, and thread through coral branches and crevices that a fast, flexible fish would have to swim around. The boxfish traded speed not for stability but for control in tight spaces, and for the freedom to be slow in the first place. That freedom depends on the second layer of defense, and it is here that the ornament becomes a weapon. Embedded in the skin of the boxfish, in the soft tissue lining the openings of the carapace, are specialized mucus glands. When the animal is seized, injured, or held under sustained stress, those glands release a secretion into the surrounding water. The active compound, first isolated from Hawaiian boxfish and named pahutoxin, is unlike the notorious nerve poisons of its relatives. This is not tetrodotoxin. Pahutoxin is a choline chloride ester of a fatty acid, a surfactant, a soap. It does not target the nervous system. it attacks membranes directly, disrupting the lipid bilayers of cells. In the gills of a fish, that means the delicate respiratory epithelium begins to fail. In blood, it ruptures red cells outright, a hemolytic effect visible in laboratory assays within minutes. The secretion foams as it disperses, and it is stable enough to survive heating, which places it in a category of chemical defenses that do not require a bite to be lethal. The consequence is that a boxfish under attack does not merely defend itself, it contaminates the water. In an enclosed volume, the effect is total. Aquarium keepers have learned expensively that a single stressed boxfish can kill every other fish in a tank, sometimes including itself, releasing enough toxin to turn a closed system into a poisoned one. On an open reef, currents dilute the discharge, but the local plume is still concentrated enough to sting the gills and mouth of whatever has just made the attempt. The lesson is delivered once, chemically, at close range, and it does not need to be repeated. This is why the boxfish advertises. Ostracion cubicus, the yellow boxfish, spends its juvenile life as a startling bright yellow cube stippled with black spots, one of the most conspicuous objects on the entire reef, before fading to a duller blue-gray mottling as it matures and its armor thickens. That juvenile coloration is aposematism in its purest form, a signal that says the cost of finding out is higher than the meal is worth. Predators that survive one encounter learn the pattern, and the pattern is honest. Across the reef, that honesty ripples outward into ecology. Boxfish are grazers and pickers rather than hunters. They move slowly across sand flats and coral rubble, blowing jets of water from their small mouths to excavate buried polychaete worms, small crustaceans and mollusks, and they browse algae, sponges, tunicates, and detritus from surfaces that faster fish pass over. Because they cannot outrun anything, they can afford to feed exposed, in the open, in daylight, on substrate that competitors visit only in nervous bursts. Their toxicity effectively removes them from the reef's food web as prey, which means the energy they consume is not being cycled upward into predators in the usual way, and it means their populations are regulated far more by habitat and disease than by predation. In the process, they act as living instruction for the reef's hunters. Every grouper, trevally, and juvenile shark that tests a boxfish and recoils carries that experience into every subsequent encounter with anything boxy, spiny, or brightly spotted, which shifts predatory attention onto other targets and creates a small, persistent bias in who gets eaten on a coral head. Warning signals that work are also warning signals worth copying, and reef systems are full of unrelated species that have converged on the same bold patterns and awkward silhouettes, borrowing protection from a reputation they never earned. So the reef repeats the lesson of the deep in brighter colors. The boxfish is slow because it can be, ornamental because ornament is a threat display, and clumsy in appearance because its survival was never staked on escape. Strip away the geometry and the yellow paint, and what remains is a small fish carrying a bone shell and a chemical weapon, hovering in plain sight above the coral, protected by the accumulated memory of every predator that ever tried. Below 1,000 meters, the last photon of surface sunlight has already been absorbed, scattered, and lost, and what remains is not dimness but absolute optical vacuum. In that vacuum, any light at all becomes the loudest possible signal. It is the only thing in the entire water column capable of drawing attention, and evolution, working on tens of millions of years of trial and consequence, has repeatedly weaponized it. Somewhere between 80 and 90% of the animals living in the open deep ocean can make their own light. Not as decoration and not as an accident of metabolism, but as functional hardware, signal lamps, counter-illumination screens, decoys, searchlights, and above all, bait. In a habitat where prey is scarce and encounters are rare, the most efficient hunting strategy is not to search, it is to advertise, and then to wait for curiosity to do the work. The chemistry behind that advertisement is deceptively simple and biologically extraordinary. Bioluminescence is a controlled oxidation reaction. A light-emitting molecule, generically called luciferin, is oxidized in the presence of an enzyme, generically called luciferase, and the energy released by that reaction is not shed as heat, but emitted directly as a photon. In the deep sea, the dominant luciferin is coelacanths, a compound so widespread across jellyfish, crustaceans, squid, and fish that many predators appear to acquire it from their diet rather than synthesizing it themselves, importing their ammunition along with their meals. The result is what physicists call cold light, with efficiencies that no filament and few engineered devices can match, converting the great majority of the reaction's chemical energy into visible output with almost no thermal waste. That matters enormously at 4 degrees Celsius under 300 atmospheres of pressure, where a heat-producing lamp would be both metabolically ruinous and instantly conspicuous to the wrong senses. The emitted wavelength is tuned as precisely as the chemistry, clustering in the blue-green band around 470 to 490 nanometers, because those are the wavelengths that travel furthest through seawater and the wavelengths that deep-sea eyes, stripped down to a single visual pigment, are built to detect. Some animals brew this light themselves in dedicated organs called photophores, complete with lenses, reflectors, filters, and shutters. Others outsource the entire process, farming colonies of luminous bacteria inside sealed chambers, feeding them oxygen and nutrients in exchange for a permanent glow they can never switch off, only conceal. No animal has committed to that strategy more completely than the anglerfish. The order Lophiiformes includes more than 300 species, and among the deep-sea ceratioid families, the females have been reduced over evolutionary time to almost nothing but a mouth, a stomach, and a fishing rod. The rod itself is the giveaway. It is not a novel organ but a repurposed one. The first spine of the dorsal fin elongated and mobilized into a flexible stalk called the elysium, tipped with a fleshy bulb known as the esca. Inside that bulb live symbiotic bioluminescent bacteria, most of them belonging to lineages such as Photobacterium and the specialized genus Enterovibrio, acquired from the surrounding seawater rather than inherited from the mother, and housed in a chamber lined with reflective tissue and served by dense blood vessels. The bacteria glow continuously. The fish controls the display not by turning the reaction on and off, but by manipulating blood flow, by opening and closing shutters of dark tissue, and by moving the entire stalk so that the light appears, vanishes, twitches, and drifts. Many escae are further elaborated into branching filaments and tendrils that spread outward from the bulb like the appendages of a small drifting animal, some of them also luminous, some of them merely silhouetted against the glow. The whole apparatus is an imitation. It mimics the size, the brightness, the flicker, and the hesitant motion of exactly the kind of small luminous organism that a deep-sea hunter has learned correctly is edible. Behind that lure hangs the rest of the animal, and the rest of the animal is a trap. Anglerfish bodies are soft, dark, low density, and almost entirely uninsulated by muscle, because muscle is expensive and the strategy demands no chasing. Their skin is often pigmented with ultra-black melanin arrangements that absorb well over 99% of incident light, so that even when the lure is blazing, the predator holding it remains a hole in the water. Species such as the black sea devil, Melanocetus johnsonii, hang motionless with the rod arched forward over the head, positioning the glowing bait a short distance in front of an enormous upward tilted mouth. The jaws are hinged for extreme distension. The teeth are long, thin, inward curving and depressible, folding backward to admit prey and locking upright to prevent its exit. The stomach is elastic enough that individuals have been recovered with prey items longer than themselves already inside. The ambush unfolds in stages, and only the last of them is fast. First comes the emission, a faint pulse of blue-green light in a void where nothing else is illuminated, drifting and stopping, drifting and stopping. Then comes the approach, a lanternfish, a shrimp, a small squid, an amphipod, anything hunting by sight in the deep, orients toward the glow, because in that environment, light is almost always attached to something worth investigating, whether a mate, a rival, or a meal. As the visitor closes, the angler adjusts, tilting the elysium so the bait drifts backward toward the mouth, walking the target into position rather than lunging at it. The prey follows the light because it cannot see the trap, and it cannot see the trap because ultra-black skin and near-zero motion leave nothing to see. Then the trigger releases. The strike is generated not by muscular propulsion, but by explosive expansion. The angler drops its lower jaw, flares its gill covers, and depresses the floor of its mouth simultaneously, and the sudden increase in oral volume produces a pressure differential that drags the surrounding water and everything suspended in it inward. In deep-sea anglerfish, this suction strike is measured in the low tens of milliseconds, some of the fastest recorded engulfments among fishes. At that timescale, the prey's own escape reflex, which requires nerve conduction, muscle contraction, and a body flick against water resistance, has not yet begun. By the time the signal reaches the tail, the fish is already inside. The jaws close, the teeth cage the opening, and the light swings back into position, unchanged, patient, waiting for the next arrival. What makes this more than a single grotesque curiosity is how many unrelated lineages arrived at the same solution. The stoplight loose jaw, Malacosteus niger, carries a suborbital photophore that emits far-red light, a wavelength effectively invisible to almost every other animal in the deep, and it possesses a chlorophyll-derived pigment in its retina that allows it to see that light. The result is a private searchlight. It illuminates prey that has no idea it has been illuminated. Its relatives among the barbel dragonfishes trail luminous chin barbels, some of them meters long relative to body size, dangled and twitched below the head as free-hanging bait. Viperfish of the genus Coelacanths carry a photophore on an elongated dorsal fin ray held out ahead of the body, drawing prey toward teeth so long they cannot be enclosed by the mouth and instead cage it from outside. The cookie-cutter shark lines its underside with photophores while leaving a dark collar unlit, and the leading hypothesis is that the resulting shape mimics a much smaller fish, luring larger predators close enough to be bitten. Deep-sea siphonophores in the genus Irina suspend rows of red glowing twitching lures along their tentacles, apparently imitating copepods, and reel in fish that have followed the flicker into a curtain of stinging cells. Even the finned octopus Storoteuthis cirtensis has converted the suckers along its arms into light organs, transforming grasping structures into a glowing net. None of these animals inherited the trick from a common lure-bearing ancestor. Fish, cephalopods, and colonial cnidarians sit on utterly separate branches of the animal tree, and their light organs are built from different tissues, powered in some cases by bacteria and in others by self-made chemistry. What they share is a problem, and the problem is identical everywhere below the twilight zone: total darkness, thinly scattered food, and no reliable way to find prey by searching. Under those conditions, the same answer keeps surfacing. Make a light. Make it small, make it move like something alive, and hold it just far enough from the mouth that the visitor commits before it understands. Curiosity in the abyss is not a flaw in the prey's design. It is a necessary survival instinct, and the hunters of the deep have spent millions of years learning to turn it into an execution. At 8,000 meters, the water column overhead presses down with roughly 800 times the force of the atmosphere at sea level. Translated into terms a surface animal can understand, that is somewhere near 8 tons bearing on every square inch of surface, the equivalent of stacking dozens of family cars onto a single fingertip and holding them there permanently, without pause, for the whole of a life. Drop past the 10,000-meter mark into the deepest trench floors, and the figure climbs beyond 1,000 atmospheres. Nothing about this environment is negotiable. Pressure does not fluctuate with weather, does not ease at night, does not relent during a storm. It is the single most consistent, most unforgiving condition on the planet, and every animal that lives in the Haddle Zone exists there because its body has stopped fighting the crush and learned instead to become part of it. The critical insight is that water itself barely cares. Liquids are almost incompressible. Squeeze seawater with 1,000 atmospheres and its volume shrinks by only a few percent. Gas is a different matter entirely. Gas obeys Boyle's law, halving in volume every time the pressure doubles, which means any air-filled space inside a body becomes a structural fault line the moment it begins to descend. This is precisely why extreme depth specialists have systematically eliminated their gas-filled cavities. The swim bladder, that elegant buoyancy organ which allows a reef fish to hover motionless over coral without spending a calorie, is absent or vestigial across the deepest living fish lineages. Where shallower relatives fine-tune a bubble, abyssal fish achieve neutral buoyancy with low-density lipids and, more commonly, with tissue so waterlogged that it barely differs in density from the ocean around it. There are no sinuses to collapse, no air spaces to implode, no compressible pockets waiting to fold inward under load. Bring a mesopelagic fish with a functioning swim bladder up in a trawl and it arrives ruined, eyes bulging, stomach everted through its mouth, destroyed by expansion rather than compression. Bring a true Haddle specialist up and it arrives intact but dying for entirely different reasons. It has nothing left inside it that pressure can distort. The real battle, however, is not fought at the level of organs. It is fought inside molecules. Extreme pressure squeezes proteins toward their most compact configuration, distorting the precise three-dimensional folds that give enzymes their function, jamming the active sites where biochemistry actually happens. It compresses cell membranes, stiffening the lipid bilayers that must remain fluid for nerves to fire and nutrients to cross. A surface-adapted enzyme dropped intact into a trench would simply stop working. Deep-sea animals answer this with a chemical countermeasure, piezolites, small organic molecules that stabilize proteins against pressure. The best documented of these is trimethylamine N-oxide, the compound responsible for the smell we associate with fish, and its concentration in fish tissue rises steadily and predictably with depth. Membranes, meanwhile, are rebuilt with a higher proportion of unsaturated lipids, a molecular adjustment that keeps them supple where ordinary fats would set. It is an arms race conducted at the scale of angstroms, and remarkably, it has a hard ceiling. Because trimethylamine N-oxide is itself osmotically active, the concentration required somewhere below 8,200 meters would push a fish's internal chemistry toward the salinity of seawater itself, breaking the osmotic balance that keeps it alive. Beyond that boundary, the arithmetic collapses. No fish has ever been reliably recorded below it. The deepest trenches on Earth, plunging kilometers further still, are a realm no vertebrate has been able to enter. Right up against that boundary lives one of the strangest survivors in the ocean, and it does not look like a monster at all. The Mariana snailfish Pseudoleperis swirei, described only in 2017, is a pale, tadpole-shaped animal rarely exceeding about 25 centimeters. Its skin is translucent, faintly pink, scaleless, and so thin that internal organs show through it. It has no fearsome dentition, no luminous lure, no armor, no speed. Its skull is incompletely calcified, its skeleton reduced and lightly ossified, and beneath the skin sits a thick layer of gelatinous, watery tissue that makes up a substantial fraction of its body. This is the key to the whole design. The snailfish does not resist pressure with structural strength, because structural strength is a losing strategy when the load reaches hundreds of atmospheres. Instead, being essentially a bag of incompressible fluid held in a flexible frame, it lets pressure pass through it evenly. Force applied uniformly to a body that contains no voids produces no stress gradients, and without stress gradients there is nothing to crack, buckle, or tear. Where a boxfish survives by building a rigid carapace to distribute a predator's bite, the snailfish survives by abandoning rigidity altogether and distributing the ocean itself, and it thrives. Cameras lowered into the Mariana Trench have filmed these fish in numbers, swimming with slow undulations near 8,000 meters, gathering around bait, feeding on the swarms of scavenging amphipods that dominate trench floors. Far from clinging to the edge of existence, snailfish are the apex predators of the Hadal Zone, and the deepest fish ever recorded on video anywhere on Earth was a juvenile snailfish observed at 8,336 meters in the Izu Ogasawara Trench off Japan, with a specimen physically collected from just over 8,000 meters in the same expedition. That footage, captured in 2022 and announced the following year, represents the current known limit of vertebrate life. The animal holding that record is soft, small, unarmored, and almost gelatinous. In the deepest habitable water on the planet, the winning body plan is not a fortress, it is a jelly. Higher up, across the vast abyssal plains between 2,000 and 6,000 meters, the same logic produces a different silhouette. Grenadiers of the family Macrouridae are among the most abundant deep-sea fishes on Earth, and species such as the abyssal grenadier, Coryphenoides armatus, occupy the seafloor in nearly every ocean basin. They have large heads, enormous eyes, and bodies that taper into a long, whip-like tail, with muscle tissue that is strikingly watery and low in protein compared with any commercially familiar fish. They cruise just above the sediment, with slow, energy-cheap tail beats, guided by extensive lateral line systems and vast olfactory chambers that read the water for the chemical signature of carrion. When a whale carcass or a fish fall reaches bottom, grenadiers arrive in numbers, sometimes within hours, drawn from great distances by scent alone. Between those windfalls they wait. Waiting is the second great adaptation, and it is every bit as extreme as the anatomical one. The abyss is cold, typically between 1 and 4 degrees Celsius, and cold slows every enzymatic reaction in a body. Deep-sea fish exploit this rather than resisting it. Their metabolic rates are dramatically depressed, their growth is slow, their lifespans are long, and their oxygen consumption is a fraction of what a comparable shallow water fish requires. Reproduction follows the same austerity. Hadal snailfish produce relatively few, unusually large eggs, investing heavily in a handful of well-provisioned young rather than gambling on volume. The reason is simple. Almost no food is generated in place. Everything edible must sink from the sunlit surface as marine snow, sunken carcasses, and drifting debris, and only a tiny percentage of surface production survives the journey. An animal living on that ration cannot afford muscle it does not need, cannot afford to chase, and cannot afford to burn energy maintaining readiness for opportunities that come months apart. Stillness is not laziness in the abyss. Stillness is solvency. Taken together, these adaptations reveal something uncomfortable about the deep ocean. Life at extreme depth is not achieved by adding capability. It is achieved by surrendering it. The swim bladder goes, dense bone goes, powerful muscle goes. Scales, pigment, speed, and the ability to sprint or fight all go, traded away for watery tissue, unfinished skeletons, and a metabolism idling barely above zero. Every one of those losses would be catastrophic in shallow water, where a fish without a swim bladder sinks, a fish without muscles starves, and a fish without pigment is a beacon. But the abyss inverts the ledger. There, the traits that define a successful surface animal are liabilities, and the animals that inherit the deepest habitats on Earth are the ones that gave up the most. Pressure does not simply kill. It edits ruthlessly, stripping bodies down to the minimum that physics will permit, and what remains at the bottom of the trenches is not a beast built to withstand the ocean, but a creature that has quietly agreed to stop being separate from it. Every predator in the ocean faces the same accounting problem. A strike costs energy, a chase costs more, and a struggle against prey that fights back can cost more than the meal returns. In the sunlit shallows where food is abundant and calories are cheap, that math can be brute forced with speed and muscle. In the dark, where a single meal might have to last weeks, brute force is a luxury few animals can afford. And so, again and again, across unrelated lineages and across every depth zone from the reef crest to the abyssal plain, evolution has arrived at the same solution. Not bigger teeth, not faster tails. Chemistry. Venom is the most energy-efficient weapon the ocean has ever produced. A gram of toxin, delivered accurately, can shut down a nervous system that would otherwise take a predator minutes of violent effort to subdue. It works while the hunter holds still. It works after the prey has already been touched only once. It converts a contest of strength into a contest of biochemistry, and biochemistry does not tire. Roughly 2,000 species of fish alone are now thought to be venomous, more than all venomous snakes, spiders, and scorpions combined, and a large proportion of them live in dim, cold, food-poor water where every other option is too expensive. Venom in the deep is not an act of aggression. It is an act of economy. The archetype sits almost motionless on the seabed. The stonefishes of the family Syringes and their broader relatives among the scorpion fishes have perfected the art of looking like nothing at all. Their skin is warty, mottled, and often overgrown with algae and sediment, breaking the outline into rubble. They do not swim so much as settle, wedging themselves into depressions, sometimes shoveling sand over their own flanks until only the eyes and the upturned mouth remain exposed. Most people know the reef-dwelling species, but the lineage extends far deeper, with scorpionid relatives such as the deep-water scorpion fishes hunting on continental slopes hundreds of meters down in permanent gloom where camouflage barely matters and the ambush is guided by vibration rather than sight. There, the strategy is identical. Wait, let the prey come. Open the mouth in a fraction of a second and inhale. The venom is not for hunting. That is the detail that reframes the whole animal. A stonefish kills its prey by suction, not by poison, and its arsenal is reserved entirely for whatever tries to eat it. Along the back stand 13 stout dorsal spines, each one a hypodermic needle with no plunger. At the base of each spine sits a pair of venom glands, and the spine is sheathed in a loose integumentary sleeve. When a predator or a human foot presses down on the animal, the spine drives upward through its own sheath, and the pressure of that compression squeezes the glands and forces venom up two grooves running the length of the spine and directly into the wound. There is no muscular injection, no fangs, no bite. The attacker supplies the force. The fish merely supplies the chemistry, which means the entire weapon system costs almost nothing to operate and can be deployed while the animal remains completely inert. What travels up that groove is a cocktail engineered for maximum disruption per molecule. The dominant component in the estuarine stonefish, Synensia horrida, is a protein called stonous toxin, a pore-forming cytolysine that punches holes through cell membranes, rupturing red blood cells and cascading into a collapse of blood pressure. Alongside it run varicotoxin and trachinyllicin, compounds that interfere with cardiac function and trigger explosive release of neurotransmitters at nerve endings, along with hyaluronidase, an enzyme whose only job is to dissolve the connective tissue matrix so the rest of the mixture spreads faster and further through the victim. The result in a human is agony severe enough to be described in medical literature as among the worst pain caused by any animal, with swelling, tissue death around the wound, and, in untreated severe cases, cardiovascular failure. In a would-be predator, the lesson arrives fast enough that the encounter is never repeated. Elsewhere in the dark, the same principle appears in entirely unrelated bodies. The chimeras, ancient cartilaginous relatives of sharks that haunt slopes and canyons down to more than 2,500 meters, carry a single erectile spine at the leading edge of the first dorsal fin, associated with venom-producing tissue, the defensive lance mounted on an animal otherwise built for slow, wing-like gliding above the mud. The stargazers of the family Uranoscopidae bury themselves in sediment with only the eyes protruding, and combine two shoulder spines sheathed in venom glands with electric organs behind the eyes, a rare pairing of chemical and electrical weaponry in a single fish. We everfishes do the same trick in colder, deeper sand. Certain deep-water rockfishes and waspfishes carry venomous fin rays as standard equipment. None of these animals is fast. All of them are heavily armed. In an environment where fleeing burns calories that cannot be replaced, standing still and being poisonous is the cheaper survival strategy. But the most surgical use of venom in the sea belongs to an animal that cannot flee at all. The cone snails of the family Conidae, several hundred species strong and ranging from reef flats down into deep water, hunt fish. A snail hunting a fish should be an absurdity, and the way it resolves that absurdity is the finest illustration of chemistry replacing muscle anywhere in the ocean. The snail extends a fleshy, flexible proboscis, at the tip of which sits a single modified radular tooth, hollow, barbed and hardened, loaded from a venom duct and fired hydraulically like a harpoon. Contact and injection happen in milliseconds. The fish is then either paralyzed where it floats or, in some species, sedated so gently that it drifts into the snail's expanded mouth without ever registering the threat. The venom itself is not one toxin, but a library. A single cone snail may produce between 100 and 200 distinct peptides collectively called conotoxins, most of them small, tightly folded, and stabilized by internal disulfide bridges that make them chemically tough and highly specific. Researchers describe them as deployed in coordinated groups. One combination, informally known as the lightning strike cabal, floods the prey's nerve endings and locks its muscles into instant rigid tetanus so that escape is impossible from the first instant. Another, the motor cabal, blocks neuromuscular transmission entirely, producing complete flaccid paralysis. Others target voltage-gated sodium, potassium, and calcium channels, or nicotinic acetylcholine receptors, each with a precision that pharmacologists find almost unreasonable. One species, the geography cone Conus geographus, has gone further still and weaponized insulin, releasing a specialized insulin variant into the surrounding water that crashes a nearby fish's blood glucose and induces hypoglycemic shock before the harpoon is even fired. That is the deep ocean's logic distilled. Do not fight the prey, edit it. None of this happens in a vacuum, and none of it is finished. Venom is one half of an arms race, and prey animals are not passive targets. Because most marine toxins work by binding to specific ion channels and receptors, any mutation that subtly reshapes the binding site on a prey animal's nerve cells reduces the toxin's grip, and such resistance has been documented repeatedly in marine systems, including in fishes and eels that are regularly preyed upon by venomous sea snakes. Resistance in the prey then selects for potency and novelty in the predator, and venom genes are unusually prone to duplication and rapid mutation, allowing whole new toxin families to be improvised over evolutionary time. The result is measurable variation in venom composition between populations of the same species living in different places and hunting different prey. The chemistry is not a fixed inheritance. It is a running negotiation rewritten wherever the opposition changes. And that negotiation now has consequences on land because deep sea and marine toxins evolve to hit single molecular targets with extreme selectivity. They make superb pharmaceutical templates, and one has already crossed over completely. Ziconotide, marketed as Frilled and approved in the United States in 2004, is a synthetic version of a conotoxin from the magician cone snail, Conus magus. It blocks a specific type of calcium channel in the spinal cord, delivering powerful pain relief without acting on opioid receptors, which means it does not produce tolerance or addiction in the way conventional opioids do. Stonefish antivenom, produced in Australia since the 1950s, remains one of the few antivenoms manufactured for a fish. Cone snail insulins are being studied for fast-acting diabetes treatments, and hundreds of other marine peptides sit in laboratory freezers awaiting screening. The animals that built these compounds did so to solve an energy problem in cold, black water where every calorie is contested. That the solutions happen to interface with human nerve cells is an accident, and it is the clearest evidence yet that the biology of the void is not a curiosity sealed away beneath the pressure. It is a chemistry set still open, and we have barely read the first page of the label. There is a kind of weapon that does not cut, sting, or poison. It simply endures, somewhere below 300 meters, in water that has barely changed temperature in millions of years. There are animals swimming today whose body plans were finalized before flowering plants covered the continents, before the great reptiles fell, before the first primate climbed anything. They are not relics in the sense of being broken or obsolete. They are the opposite. They are designed so thoroughly matched to a stable, sunless world that natural selection has found almost nothing worth changing. Biologists use the term living fossil with some discomfort because these creatures have not stopped evolving. Their genes mutate, their populations shift, their chemistry adapts. What has frozen is the shape, and that frozen shape is itself an achievement, because every other branch of their family tree, Every cousin that experimented with new jaws or new fins or new hunting grounds nearer the surface has been erased. Longevity of lineage is a survival strategy in its own right, and the deep ocean is where it is practiced best. Consider the frilled shark, Chlamydoselachus anguineus, an animal that looks less like a shark than a mistake. Its body is elongated and eel-like, reaching around 2 meters, brown and soft-skinned, with a single small dorsal fin set far back near the tail. Its head ends not in a shark's usual undershot snout, but in a mouth positioned right at the tip, opening forward like a trap. Behind that mouth sit 6 pairs of gill slits, the first pair running continuously across the throat, their fleshy margins fringed and ruffled, which is where the name comes from. Most modern sharks have 5. Inside the jaws are roughly 300 teeth arranged in some 25 rows, and each tooth is a needle-sharp trident of 3 backward curving cusps, gleaming white against dark tissue, apparently visible enough in dim water to attract curious prey directly into the strike zone. Its spine is barely a spine at all, retaining a largely unconstricted, fluid-filled notochord where later sharks developed hardened, segmented vertebrae. This is skeletal architecture from an earlier chapter of vertebrate history still in service. The frilled shark's lineage stretches back through the Chlamydoselachidae into the Cretaceous, and its relatives appear in fossil beds tens of millions of years old, with teeth almost indistinguishable from those in living specimens. It occupies the outer continental shelves and upper slopes, most often between about 120 and 1300 meters, and it is encountered so rarely that most of what science possesses comes from bycatch hauled up dead in deep water nets, or from the occasional dying individual that drifts, disoriented, into shallow water where a diver or fisherman films it, and the internet briefly loses its composure. Its reproduction is as archaic as its anatomy. Embryos develop inside the mother without a placental connection, nourished by yolk, and the gestation period has been estimated at around 3.5 years, among the longest known for any vertebrate. Nothing about this animal is fast except the strike, and the strike is the point. With a terminal mouth, jaws that can open extraordinarily wide, and a flexible muscular body, the frilled shark is thought to hunt by lunging forward like a snake, bending its body and springing at soft prey, mainly squid, which dominate its stomach contents. Those trident teeth are not built for cutting flesh, they are built for snagging. Angled backward and interlocking, they function as a one-way gate, hooking into the slick mantle of a squid so that every attempt to pull away drives the barbs deeper. Some researchers suspect it can also close its gill slits to generate suction, drawing prey inward as the mouth opens. It is a primitive mechanism crude by the standards of a great white or a mako, and it has never needed refinement, because in a lightless world where squid drift within reach, a snake-like lunge and a mouthful of hooks solves the problem completely. Evolution does not improve what already works. Yet the frilled shark is not the most staggering survivor down there. That title belongs to a fish that science had already buried. The coelacanth was known only from fossils, a lobe-finned lineage traced back some 400 million years to the Devonian, and the record appeared to stop dead around the end of the Cretaceous 66 million years ago. Then, in December 1938, a trawler working near the mouth of the Columna River off South Africa landed a large steel blue fish with heavy scales and strange fleshy limbs. Museum curator Marjorie Courtney Latimer recognized that she was looking at something impossible, and ichthyologist J.L.B. Smith later confirmed it. The species was named Latimeria calumnae. Almost 60 years afterwards, in the late 1990s, a second species, Latimeria menadoensis, was identified from Indonesian waters more than 9,000 kilometers away. Two populations of a supposedly extinct order hiding in plain sight in fishermen's nets for as long as fishermen had been casting them. Its anatomy is a museum of abandoned engineering. Instead of a segmented backbone, the coelacanth retains a thick, oil-filled notochord running the length of the body. Its skull is hinged by an intracranial joint, a mobile break in the brain case that allows the front of the head to swing upward and widen the gape, a feature found in no other living fish. Where other fishes carry a gas-filled swim bladder, the coelacanth carries a long fatty organ saturated with oil, incompressible and therefore untroubled by pressure at depth. In its snout sits a rostral organ, a gel-filled cavity that detects the faint electrical fields generated by living muscle, allowing it to locate prey hidden in rock crevices in total darkness. And then there are the fins. 7 of them, thick and stalked, mounted on bony, muscular lobes, and when the fish swims slowly, they move in a coordinated diagonal pattern, left front with right rear, the same alternating rhythm used by a walking salamander. This is not a fish that resembles our ancestors by coincidence. It preserves the mechanical logic that eventually carried vertebrates onto land. Both coelacanth species live along steep volcanic slopes, sheltering by day in submarine caves at depths of roughly 150 to 250 meters, and drifting out at night to hunt, often head down, cruising the current and letting prey come to them rather than chasing anything. Their metabolism is exceptionally slow, and a 2021 analysis of growth rings in their scales revised their biology dramatically, suggesting a lifespan approaching 100 years, sexual maturity delayed until around the age of 55, and a gestation period that may last 5 years. It is the exact inverse of the surface world's strategy. No speed, no fecundity, no gamble, just persistence. The reason both these animals still exist while their entire families vanished has less to do with superiority than with address. The catastrophes that reset life on Earth struck hardest at the surface. When an asteroid ended the Cretaceous, the immediate collapse came in the sunlit layer where photosynthesis failed, plankton communities crashed, and the food webs built on them, including the ammonites and the great marine reptiles, disintegrated within a geological instant. Deep water buffers everything. Temperature there varies little, light never varies at all, and the animals living on scavenged organic matter and slow-moving prey were insulated from the worst of a sudden, violent shock. Add low competition, minimal environmental volatility, and metabolisms tuned so far down that a single meal can be stretched for weeks, and you have a refuge where an ancient design faces no pressure to modernize. Stability does not reward innovation. It rewards conservatism, and it punishes the experimental. What survives, then, is scientific evidence of a kind that cannot be reconstructed from stone. When the coelacanth genome was sequenced in 2013, it revealed unusually slow rates of change in its protein-coding genes, consistent with a body plan under long-term stabilizing selection, and it helped clarify how the genetic toolkit for limbs and air breathing was assembled in the water, long before anything crawled out of it. The frilled shark offers a comparable window into the early architecture of shark jaws and vertebral columns. Alongside them swim other survivors of nearly emptied branches, the goblin shark with its slingshot jaws, the nautilus with its lensless pinhole eye and gas-regulated shell, the vampire squid, sole living member of an entire cephalopod order, gathering falling detritus with a sticky retractable filament rather than hunting at all. They are the last representatives of evolutionary roots closed everywhere else on Earth, and they are fragile precisely because they are so few. The West Indian Ocean coelacanth is listed as critically endangered, with perhaps a few hundred individuals around the Comoros, and its greatest threat is the deep-set gillnet, dropped by fishermen targeting oilfish in exactly the depths where the fish shelters. 400 million years of continuity, riding out asteroid impacts and mass extinctions in the dark, and the modern hazard is a curtain of nylon. The deep sea protected them from everything the planet could throw. It cannot protect them from us. Strip away the names, the taxonomies, the depths, and the dates, and every animal in this catalog is answering the same interrogation. The deep ocean asks each of its residents a single question, repeated without mercy across 4 billion cubic kilometers of cold black water. How do you stay alive where there is no light, almost no food, and a weight of water pressing in at more than 1,000 atmospheres? Every creature profiled here is a different sentence in the same answer, and when you lay those answers side by side, the separate marvels stop looking like curiosities and start looking like variations on one ruthless design philosophy. Transparency was the first solution, and it is the most elegant. The glass squids of the family Cranchiidae did not evolve invisibility for beauty. They evolved it because in the twilight zone between 200 and 1,000 meters, where the last blue photons filter down from above, a silhouette is a death sentence. Tuning tissue refractive index to match seawater, flooding the body cavity with buoyant ammonium chloride, hiding the digestive gland and screening the eyes behind counter-illuminating photophores, all of it exists to erase a shadow. Invisibility in that water column is not a trick. It is armor made of nothing. Where light is abundant and space is contested, the calculus reverses and animals build outward instead of vanishing. The boxfish carapace, its hexagonal scutes fused into a rigid shell that spreads the force of a bite across the entire body, is the shallow water inversion of the same problem. And even that plating is not the real weapon. The real weapon is phytotoxin seeping from the skin, a surfactant that ruptures cell membranes and can poison an entire enclosed volume of water, advertised in advance by colors so bright that predators learn them once and never test them twice. Defense and offense collapse into a single organ. The shell announces the poison. The poison justifies the shell. Below the last of the sunlight, animals stopped hiding from light and began manufacturing it. Cholinterazine and luciferase, burning cold at 470 nanometers in perhaps 80 to 90% of the animals in open deep water. The anglerfish carries its lure ahead of its face, a bulb of symbiotic bacteria dangling above jaws that distend beyond all reason, its skin engineered to absorb over 99% of the light that touches it so that only the bait is visible. The stoplight loosejaw broadcasts red, a wavelength most of its neighbors cannot see, and hunts with a private searchlight. Light there is simultaneously a fishing line, a camouflage system, a burglar alarm, and a mating call. Nothing in the abyss is ever used for only one purpose, because nothing down there can afford a specialist tool that does a single job. Deeper still, adaptation becomes subtraction. The Mariana snailfish does not resist pressure, it surrenders to it, abandoning the swim bladder, the dense bone, the heavy muscle, the pigment, until what remains is a translucent gelatinous animal that transmits force evenly through itself and therefore has nothing left to crush. Its cells are packed with trimethylamine N oxide to hold proteins in shape, and it is that molecule, not courage or engineering, that draws the hard ceiling at roughly 8,200 meters below which no fish has ever been found. The abyssal grenadier applies the same economy across the vast plains between 2 and 6,000 meters, running a metabolism so depressed that a single carcass drifting down from the surface can fund weeks of existence. Survival at depth is not accumulation. It is the systematic amputation of everything that costs energy and buys nothing. And when a body cannot outrun, outgrow, or outfight its neighbors, chemistry does the work. Venom is the poor animal's arsenal, the strategy of the ambusher and the buried, of the stonefish lying under a crust of algae with 13 loaded dorsal spines, of the cone snail firing a hollow radular tooth into a fish and flooding it with hundreds of conotoxin peptides, some shutting down nerve conduction in seconds, others delivering weaponized insulin to collapse the prey's blood sugar before it can even flee. A gram of venom does the work of a body 10 times larger. In an environment where calories are the hardest currency on Earth, that ratio is everything. Then there are the animals that solved the problem so long ago that they simply stopped changing. The coelacanth, presumed dead for 66 million years until a trawler off South Africa proved otherwise in 1938, drifting nose down through submarine caves with a lifespan approaching a century. The frilled shark, with its notochord and its 300 backward curving trident teeth, striking like a snake. They persist not because they are superior, but because deep water is the only address on this planet where the bill for evolutionary stagnation never arrives. When the surface burns, the abyss barely notices. 6 strategies then, but only one logic. The deep ocean functions as the most severe filter biology has ever been run through, and what it filters out is versatility. On a coral reef, in an estuary, in a kelp forest, a generalist thrives because conditions change and opportunities multiply, and the animal that can eat many things and occupy many niches wins. Below 1,000 meters, conditions never change and opportunities almost never arrive. Under those terms, flexibility is dead weight. The filter rewards the animal that has abandoned every capability except the one that keeps it alive, and it deletes everything else with the patience of geological time. That is why the deep sea produces creatures that look impossible. They are not designed for a wide world. They are honed to a single point, and outside that point, they cannot exist at all. Haul a snailfish to the surface and it dissolves. Bring a glass squid into daylight and it becomes an ordinary, obvious animal, suddenly and fatally visible. Which brings the argument to its harder edge. In this environment, danger and survival are not opposites. They are the same phenomenon observed from 2 sides. The anglerfish lure that attracts a meal is also the beacon that reveals the anglerfish. The transparency that conceals the glass squid also renders it structurally fragile. The boxfish carapace that stops a bite also makes the animal a poor swimmer, dependent on constant corrective sculling to hold a line. The stonefish spine that kills a predator cannot be used to hunt at all. Every adaptation examined here is a weapon, a shield, and a liability fused into one, purchased at a price the animal pays every single day of its life. That is the closing verdict. The deep ocean does not produce survivors. It produces specialists so extreme that survival and vulnerability become indistinguishable. And the honest conclusion is that we have barely begun to read the evidence. More than 80% of the ocean has never been explored, mapped, or observed by any human instrument. As of the early 2020s, only around a quarter of the global seafloor had been charted at modern high resolution, meaning the topography of most of our own planet remains less understood than the surface of Mars. Roughly 2,000 new marine species are formally described every year, and credible estimates suggest that 2/3 of all ocean life still has no scientific name. Hydrothermal vents, entire ecosystems running on chemosynthesis rather than sunlight, were not discovered until 1977. The barrelized, transparent, fluid-filled head was not properly understood until it was filmed alive in 2004. A luminous, hood-feeding deep-sea mollusk entirely new to science, Bathydevius caudactylus, was formally described only in 2024 after 20 years of sporadic sightings. Worse, the traditional net drags gelatinous animals up as unrecognizable slurry, meaning some species have almost certainly been destroyed before they were ever recognized. So every animal in this catalog should be understood as a single recovered page from a library we have not opened. The transparent hunters, the armored chemists, the light bearers, the pressure-adapted ghosts, the venomous ambushers, the survivors of vanished ages. Each one is a fragment, and the pattern they trace points inward, downward, toward water no camera has yet reached. The rarest animals on Earth are not the ones we have filmed. They are the ones still holding position in the trenches and the seamount flanks and the abyssal plains, refining strategies we cannot yet imagine. That is the case for going back. Not curiosity alone, but the certainty that the most efficient predators, the most surgical toxins, and the most extreme biological engineering this planet has produced are almost certainly not in our collections yet. They are down there in the cold and the crushing dark, waiting, because nature keeps its most ruthless designs precisely where the light has never reached.